Good afternoon, folks, and welcome to your final lecture. How about that?
Woohoo!
Okay, welcome back. And thanks for coming out. I know it's hard to stay energized when you're exhausted and you just kind of want things to finish. So, just try your best to power through here to get through your final exam next week. And then you hopefully, at least from this class's perspective, can kind of collapse and take a break. Okay, so let me just get through a couple business items here. Of course, you know that your final exam is a week from today, Thursday, December 16th at 12:30. Thursday, December 16th at 12:30 is your final exam. Okay, so let's open the, we have a lot to do in terms of trying to cover major topics of chapter 17, but I do want to start today by asking you, is there content, are there questions for the final exam that you would like some guidance on?
I just was wondering, are you going to have office hours even though we don't have class?
Okay, so thanks for asking, Samantha. No, I will not be holding office hours next week. Okay. I will not be holding office hours next week. And so, but I will be monitoring email on a daily basis. So, if you do need me, if you want to reach out with a question about something, I will get back to you. I mean, don't send me the question Wednesday night at 11 o'clock, right? That's going to be hard for me to turn that around in time to help you, but I will respond to emails next week. So no office hours going into next week, but you can still reach out to me through email.
Okay, thank you.
Let me just take a look at the chat windows here. Okay, Kit, you're asking, are we getting any more detail on the study guide portions that say thought questions from chapter 15 or chapter 17? No, Kit, you're not getting any more detail, okay? But I will say this. I firmly believe that the final exam is less intense of an exam than exams 2 or 3. So I want you to study, and if I think if you study and prepare, you read the chapters, you look over the notes, review the lectures we've given, have your brain turned on, right? So your mind is active, you've eaten a meal before the exam, so you're not running low on sugar. You're going to be just fine, okay? You're going to be just fine. Okay. Okay. So, I'm looking at a Falashati on the exam guide. Thought question 17 appears 2CE. I'm not sure what you're trying to. I'm not sure what you're trying to tell me, Falashati. Oh, okay. Is it on there twice? Okay.
Yes, it is.
Okay. All right. Okay. Thank you for saying that. Let me just take a look at that study guide here while I have you, make sure it's okay. Excuse me while I go through some files here. Oh, okay. All right. Yes. 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 Okay. I see what you're telling me. So let me bring this up. Okay. So on the study guide, essay number 3 says it's going to be a thought question from a topic in chapter 17. And down here, I have essay 7, a thought question from chapter 17. And this is correct. What I'm telling you is there will be 2 essays where I challenge you to think from content in chapter 17. They're 2 different questions, but they're both from chapter 17. Okay. All right, let's see here. Okay, so Samantha, your comment, the questions in the back of each chapter, the chapter review really help. Yep, I agree. That is something that you should spend time on. Okay. All right. Okay. So am I missing anything in terms of chat window that you want me to cover? Please speak up. I don't think I'm missing anything. Okay. All right then, gang. So let's continue here. And let me just take a quick look. You know, we're not going to have a traditional lab today. However, it is possible that I might have to use a little bit of time in lab for lecture. We'll see how it goes. But before I forget, let me give you all your latest lab homework, the Nipah virus work that you did. Let me give you your grades. And so this will be fairly easy because just about everyone earned 100%. Just about everyone earned 100%. And so let me just take a quick look at who I have in attendance here. Okay, so bear with me. I want to make a communication to one person. Okay, so I've communicated with one person in this group. And so now I'll tell you, if you did not get a message from me, you earned 100%, you earned full credit on your Nipah lab activity, okay? So most of you did just fine with that. Doesn't mean you got perfect. It doesn't mean everything was 100% correct. It means I'm satisfied with the work that you did provide to me. Okay, so in terms of moving forward, obviously, you know, you still have the immunology lab, which I haven't graded at all yet. And so, you know, if you're burning to know what your score is on that homework, you know, sometime next week, shoot me an email. After the final exam, I will, you know, next Thursday after the final exam, once I have the exams graded and finished, I will send out a traditional class-wide email letting you know that your grades are available, okay? So until you see that email from me, your exam grades are not complete, okay? They're not finished. So there'll be 1 or 2 more emails coming out for me. So just, you know, try to stay alert for that as we finish up the semester.
Will you give us an individual grade with the class marker, or is that we just check on that once it's official? And then you'll send us the overall grade for the semester?
Okay, so Elena, once the exam is finished, I'll communicate to you that the exam scores are ready for you to get, so you can log in the class marker to get your exam scores. I will be calculating your overall course grade using the method that I've shared with you a few times this term in that Excel spreadsheet. And I'll be entering that information into the HACC database, okay? It is up to the HACC administration when to release those grades. So I would anticipate, so your final exam is Thursday the 16th. I doubt very much that HACC's administration will release course grades any earlier than Monday afternoon on December 20th. So you could look in your MyHack account on Monday to get your course grade whenever that's released. Honestly, if you email me using your Hawkmail account, it has got to be your Hawkmail account for legal reasons, then I would respond with your course grade for you. Okay. Okay. All right, gang. Okay, so anything else, any other business items we should talk about? Okay, so let's see what we can do in chapter 17.
I have one
Okay.
question quick.
Okay,
So do we not have class on Tuesday?
that is correct, Rachel. There is no class on Tuesday. So
Okay.
it'll be a week, between today and next Thursday. It'll be a week until you're asked to formally do anything with micro again.
All right, sounds good. Thank you.
Mhm. Okay, anything else? Okay, let's continue. Okay. Okay, so chapter 17 is about adaptive immunity. So, if any of you have had immune system topics in the past, you know that the immune system is really not the whole story when it comes to body defense. And if you've had A&P, you know that to be true. In fact, in your own microbiology textbook, chapter 16 is about innate immunity, and chapter 17 is about adaptive immunity. Well, why aren't we covering both chapters? Well, gang, because we're just out of time. That's why we're not covering both chapters. Okay, so hopefully in another course that you'll take, you'll get a more complete, holistic coverage of this topic than you're going to get in one lecture here in microbiology. But let's just talk very quickly about an overview of body defense. Okay, you can see in the note-taking document that I have 3 video links up here for videos that I will recommend to you during your study time. I do anticipate showing you 2 of the videos today in class, and the third one, if you're interested, you can do on your own time. Okay, and so when we talk about body defense, please understand that body defense is most definitely not, it is not synonymous with immune system. Immune system is only a component of body defense. So let's put in a table here, a 2-column table, where I'll put a couple items in, not sure how much I'll need. In the first column, this would be the innate defenses, and this would be the topic of chapter 16. I'm only referencing it for your notes. I'm not requiring that you read it. You do not have to read it. I will not go out of my way to pull something out of chapter 16 on your final exam. I'm not going to do that. What I'm telling you is to try to give you a bird's eye overview now so that we can have a meaningful conversation about chapter 17. I should at least make some preliminary summary statements here. So chapter 16 is the innate defenses. When we think about innate defenses, we should be thinking about defenses that are non-specific. Okay. And so when you ask yourself, well, what does non-specific means? It means that the defense is capable of handling a threat without knowing the identity of the threat. The defense is capable of defending you without knowing the identity of the threat. Okay. Okay. And what we'll be spending most of our time is in the adaptive defenses. Okay, this is chapter 17, which is assigned reading. Okay, and adaptive defenses are specific. And by specific, I mean that the body is able to mount a customized response to a threat because the body can generate specific cells to defend itself because it can identify the threat. Okay. Now, your body cannot identify a pathogen and say, that's E. coli, that's coronavirus, that's HIV, that's Vibrio cholera. Your body cannot do that. It's not going to put a name on the pathogen like you and I put a name on things, right? Oh, that's Bob walking across the street. That's Sally shopping for groceries. Your body doesn't do that. But in an analogous way, at the molecular level, the cells of your body can identify things. And that's what's happening with the adaptive defenses, which is our talking point today from chapter 17. So specific means the body can customize a response to a threat because the threat can be identified. Okay. All right. So in terms of innate defenses, which again is not, it's not a talk, it's not a major theme of today, but I simply want to give you an overview here in 5-10 minutes. When we talk about innate defenses, that would include things like physical barriers. Okay. And when you say physical barriers, probably your skin comes up as an example, right? Your skin is a physical barrier. Your skin is the most exterior surface of the body, and it will block a variety of microorganisms in the air and so forth that want to enter your body, right? So the skin is a physical barrier. So the skin is going to block a variety of different species of microbes, whether it's E. coli, Salmonella typhimurium, Mycobacterium tuberculosis, whatever it might be, the skin doesn't care what the identity is, it simply blocks. It's kind of like the wall of your home. The wall of your home keeps out snow, keeps out rain, keeps out wind, keeps out a burglar. Okay, it doesn't matter what the threat is, the wall simply blocks it. So skin is a physical barrier. Also, your mucous membranes are going to fall into that place as well, right? Mucous membranes throughout your body are physical barriers. We also have chemical barriers, which, will block things in a non-specific way. And here I'm thinking about things like your skin oil, right? You learned in this course that your skin oil has mildly acidic pH, which gives the skin oil antibacterial properties. Okay. Other chemical barriers would include liquids in your body like saliva in your mouth, right? Saliva in the mouth has antibacterial molecules in it. Okay. So this is just, a way to start thinking about how your body's anatomy is designed to be protective for you. In terms of some additional innate defenses, we would include other things like various categories of defense cells. So that would include things like macrophages. You probably know about macrophages. We're going to talk a little bit about macrophages, okay? But macrophages would be an example of a defense cell. There's all kinds of other ones as well. There are also categories of defense proteins. Okay, when I'm talking defense proteins, two major categories that come to mind are complement proteins and interferon. Okay. so I will continue here. Let's see, two more things I want to mention. And that would be inflammation. Inflammation is considered a non-specific defense. You know, I'm sure we've all had inflamed areas of our body. You might have had an inflamed fingernail. You might have had a hangnail coming off the edge of your fingernail and it might have been inflamed. You might have had an inflamed pimple. You might have had an inflamed cut on your arm or hand or toe. Who knows what it is, right? You might have had an inflamed tooth. I mean, inflammation is an innate defense. There's a whole bunch of symptoms that are associated with inflammation, right? Such as redness, swelling, pain, that kind of thing. And then there's also the non-specific defense of fever, right? When your body elevates its overall temperature, that is a non-specific defense. Okay, fever basically works by causing your body to hold on to more heat energy than it normally would. So your body temperature goes up. So that means any pathogen that's inside you, whether it's a virus, a bacterium, a fungus, protozoa, that pathogen has to survive in a hotter environment than it normally would. And therefore, hopefully it won't survive and it will die, or at least not work as well because of the increased body temperature. So these are examples of non-specific defenses that are still very powerful ways that your body has to keep you safe, right? And I mean, I'm sure every one of us at some moment in our life has had a fever, so we know exactly what that is. I mean, we've experienced it. So unless you have questions, I'm not going to really talk anymore about that. And I'll talk to you now about the content of chapter 17. And when we talk about adaptive defenses, we are talking about typically different lineages, different types of cell categories that are capable of specializing for defense, okay? So these are typically going to be your B lymphocytes and your T lymphocytes. These are sort of the master warriors of your adaptive defense line. The B and T lymphocytes are the master warriors of your immune system from a proper perspective, okay? These cells can adapt to a threat because at the molecular level, they can recognize the threat, okay? So one last thing, and I'll put this in both columns. It's important to think about that even though textbooks might talk about innate defenses in one chapter and adaptive defenses in a second chapter, your body doesn't think that way. In other words, the cells that are part of innate defense have the ability to communicate with the cells of the adaptive defense and vice versa. So there is tremendous crosstalk between innate and adaptive defenses, okay? There is tremendous crosstalk on both sides of the aisle. Both sides of the aisle are communicating with each other, okay? And so let me just put it this way. I'm going to put that same statement here, and I'm just going to put this in red font so you see it, okay? There is crosstalk between these two different categories of defense, okay? It's not like Democrats don't talk to Republicans. That doesn't happen in the body, okay? At least it's not supposed to happen in the body. Okay, so there's a lot to do here. And what I'm going to do is start by showing you an overview video of your immune system, so the adaptive defenses, and use that to tickle you with some questions. And then what we're going to do is step through some basic scenario of how your immune system might respond to a threat in the body, okay? So let's get the first video started here. I'm going to cue it up. Okay, and here we go. All right. First video, the immune response. This is just a general overview for those that have really had no exposure to this content. Here we go.
Immune response. The immune system protects the body against invaders, such as bacteria and viruses. The skin serves as an important first line of defense against foreign invaders. The breaching of the skin, for example, by a splinter, could provide a route for bacteria to enter the body. These invading bacteria are regarded as non-self substances by the body's immune system, thus triggering an inflammatory response in an attempt to destroy the invading agents. A special type of WBC, known as a macrophage, engulfs the non-self bacterium, reducing it to tiny remnants known as antigens, which are displayed on the external surface of the macrophage, thus serving as an antigen-presenting cell or APC. This alerts the body's other macrophages to the presence of bacteria in the body. The macrophage also communicates with these cells by releasing cytokines, which fit snugly into the receptors of the cells, much like a lock-and-key mechanism. This results in the increase in the number of macrophages at the site of the inflammation. Additionally, some of the normal cells also function as antigen-presenting cells by ingesting some of the bacteria and displaying the antigens on their surface. This type of immune response is immediate and non-specific and is known as innate immunity. However, as the macrophages cannot destroy all the invading bacteria in the body, they proceed to the peripheral lymphoid tissues, such as the lymph nodes, in order to alert other immune cells to the inflammatory response taking place in the body. As lymph nodes function as a gathering place for several of the cells of the immune system, the macrophages are able to activate the T and B cells, which are different types of WBCs, which are then able to mount an immune response. This type of immune response is specific, and as it takes the body a longer time to mount such a response, it is known as acquired or adaptive immunity. T helper cells are stimulated by interleukin-1, secreted by the antigen-presenting macrophages, and in turn secrete interleukin-2, which help in the activation of other immune cells, especially cytotoxic T cells and B cells. Cytotoxic T cells bind to the antigen on the antigen-presenting cells and secrete chemicals, which in turn destroy these cells. This type of adaptive immunity involving the cytotoxic T cells is known as cell-mediated immunity. Activated B cells could be of two types: plasma cells or memory B cells. Plasma cells secrete antibodies that bind to the antigens on the surfaces of the antigen-presenting cells or bacteria, which are then destroyed by the macrophages. This type of adaptive immunity involving the plasma cells and antibodies is known as humoral immunity. The memory B cells, along with the memory T cells, survive for several decades and mount a secondary immune response to a future infection by the same pathogen. This same principle is employed during the vaccination of children and adults against common infectious pathogens.
Okay, folks, so sorry about some of the kind of random ads that pop up there. That's kind of a thing that happens when you're using free content on YouTube. But nonetheless, the story of the immune system was pretty well done. So we're going to have to talk about this. Do you have any questions at the moment? Okay. All right, any questions you'd like to ask from having seen the video one time? Okay, all right. All right, let's continue then. Okay. Okay, so with that video in your mind, now you have a little bit of way to think about things as we're going through this conversation. Okay, so let's talk about adaptive immunity, your immune system. Okay, so just a tiny bit of background here. Okay, so we've already mentioned up here in our table that the B lymphocytes and T lymphocytes are the major warriors of the adaptive defense system. And while it seems like there's only two types of cells, B cells and T cells, the reality of it is there are many subtypes of B and T lymphocytes that get involved in the true immune response, most of those which we're kind of just going to gloss over for our conversation right now. Okay, so so I would refer you to figure 17.1. It shows you where the B and T cells originate, where they come from. So lymphocytes are manufactured in your lymphocytes are manufactured by stem cells that exist in your red bone marrow. Okay, lymphocytes are manufactured by stem cells that exist in your red bone marrow. So both B lymphocytes and T lymphocytes originate in your red bone marrow. Okay. So to break it down a little bit more, so B lymphocytes, which you'll probably hear me say and type out into the future, B lymphocytes are often known as B cells. So if I say B cell, I'm talking about a B lymphocyte. Okay. The B cells are born in the red bone marrow, and they stay in the bone marrow for a special training process that we'll get into a little bit further down the road. Okay. So they are born in the red bone marrow and stay there for training. Okay. The T lymphocytes, or T cells, if you will, makes it a little bit shorter to say. Okay. They are also born in the red bone marrow, but then they actually physically migrate out of the red bone marrow for their training. So the training of the T cells is a little bit different from the training of the B cells. Okay. And born in the red bone marrow and travel to the thymus, which you see that in figure 17.1 for training. Okay, you see in figure 17.1 that word differentiation. And for those that know what that is, basically that means that the cells start to specialize through the differentiation process. When cells specialize, they're differentiating. Differentiation is always coupled with a change in gene expression. Okay, so for a B cell to specialize into a different subtype of B cell, there has to be an alteration in gene expression. Okay. All right. So, okay. So next, I have a question for you, and I'm hoping you can answer it for me. There's, to talk about the adaptive immune system, we want to consider some points that have a little bit of a philosophical nature. Okay. Here's the question in your notes. What is the first thing that a B or T cell must be capable of doing before it can adapt to a threat? What would you say the answer to that question is? Please contribute in the chat window. Okay, I'm just going to wait for another 1 or 2 responses, and I appreciate Kit and Elena, you responding so quickly. So see if you can contribute, folks. What do you think about this? What is the first thing that has to happen? Okay. Any other thoughts out there? Okay, well, to keep our conversation moving, since we are low on time, I will say that, you know, I very much like the idea of identifying the threat. I mean, think about it. How could you respond to a threat in your home if you don't know what the threat is? You know, if there's somebody in your house, how are you going to defend yourself unless you can recognize that there is somebody in your house? I want to say another comment, which is very subtle distinction. And maybe I'll put this in the chat window. Here's what I'm going to put in the chat window. Okay. So imagine for a second, folks, that we had a normal semester and we're sitting in a classroom together. Okay. Imagine that we're all sitting in a physical classroom together. And from that perspective, I want you to consider my next question. If someone you have never seen in our physical classroom suddenly enters our room today, would you consider that person foreign? Okay. All right. So if you're saying yes, then that is the perspective that I would want you to think about this. And that's good. So, you know, we've been together all semester. We're sitting in the classroom. Somebody enters that we've never seen. Aren't you going to say, huh, never seen that person before? That person's foreign to me. I don't recognize that person. Now, stay with me here. Another question in the chat window. Here we go. Is knowing that the person who entered the room is foreign to you different from knowing the name of the person that just entered the room? Okay. Do you see the subtle difference there? Do you see that you could have somebody in the room that you've never seen before, so that person's foreign, but simultaneously you do not know that person's name. So you know the person's foreign, but you cannot identify the person. Okay, there's a very subtle and important distinction here. Recognizing that something is foreign is different than identifying the foreign thing. Okay, Okay. So let's get some of these ideas down in your notes in relation to this question here, okay? So what is the first thing that a B or T cell must be capable of doing before it can adapt to a threat? I agree with you that the B or T cell must be capable of identifying the foreign thing. Must be capable of identifying the threat. And so we appreciate here that there is a very subtle distinction between recognizing that something's foreign and identifying the foreign thing. There is a subtle but deeply important distinction between recognizing something as foreign and identifying the foreign thing. Okay, you might want to read that a couple times during your study. There is a subtle but deeply important distinction between recognizing something as foreign and identifying the foreign thing. And so because we only have today to talk about this, I'm just going to kind of feed you what I want you to know. I mean, normally I would ask you to wrestle with it on your own a little bit, but I'm not going to have time to help you more along the way after today. So with this idea in mind, it's important that you appreciate that recognizing that something is foreign in the body typically happens by cells that are part of the innate defenses, particularly the macrophages. The macrophages are very good at identifying things that don't belong, but they're not good at identifying the identity of the thing that doesn't belong, okay? Macrophages of the innate defenses are good at identifying foreign things. And I'm using that term things loosely, right? It would be a foreign bacterium, a foreign virus, foreign protozoa, that kind of thing. Okay. Whereas B and T cells of the adaptive defenses are good. They are good at identifying. Let me change my language in the notes because I want to be clear to you. Macrophages are good at recognizing foreign things. Okay, recognition is not the same thing as identifying. B and T cells are good at identifying foreign things. But remember, okay, okay, trying to talk and type at the same time and do a good job is hard here. So macrophages are good at recognizing foreign things. They recognize that something doesn't belong. B and T cells of the adaptive defenses are good at identifying foreign things. So Yasmin, to your question in the chat window, and I'll just read Yasmin's question. Yasmin says, so the B and T cells would know the name of that bacteria? Yes, they wouldn't know that it's E. coli, but they would know the molecular fingerprint of E. coli. So yes, Yasmin. Okay. And so there's one other subtle thing that I'm going to plant into your mind at this time, and that's the following. Okay. The B and T cells, which are good at identifying foreign things, they are better able to do their job when they communicate with the cells that are good at recognizing foreign things, which are the macrophages. Hence, the idea up here of crosstalk between these two different categories of defense. To get a complete picture, well, we need more time, and we're just not going to have that luxury today. Okay. Are you with me right now, folks? Am I making sense? Okay, good. Thank you. Folks, let me say one thing. I do have a sense that, from the study guide that I put forth for this final exam, some of you might be thinking, wow, he didn't really tell us anything. That was a bit intentional. But I will tell you this. The answers for the chapter 17 essay questions, which I gave you no hints on in the study guide, they're going to be covered in today's lecture. So it's very important that you're paying attention to today's lecture, okay? So let's continue. Okay, B and T cells are best at their job when they communicate with the macrophages, okay? All right, let me just look here. I want to be sure that I'm giving you a readable document here. So my question was, what is the first thing that a B or T cell must be capable of doing before it can adapt to a threat? And it is true that it must be capable of identifying the threat. It goes a long way for the B and T cell to identify the threat if it can communicate at a chemical, molecular level with the macrophages, okay? So, let me ask you another question. Okay, so for a B or T cell to recognize an enemy, it has to be able to, for you and I, as human beings, to recognize each other, all we have to do is look at each other, right? You look at me and say, okay, that's my micro teacher. I look at you and I say, okay, that's my micro student. Or you look at your aunt and uncle and you see them and you know their names immediately, right? But you understand that cells in your body do not have eyeballs. They don't have eyeballs. They also don't have ears to hear sound waves, things like that, right? But what cells of our body do have are receptors that allow them to bind other molecules. Okay, they have receptors to bind other molecules. So let me make a statement here and then I'll follow it up with a question. In order for B and T lymphocytes to adapt to a threat, they must identify it. Identification requires that a molecule on the surface of the lymphocyte touches a molecule on the surface of the pathogen. Okay. Identification requires that a molecule on the surface of the lymphocyte touches a molecule on the surface of a pathogen. Okay, it would be kind of like, and I don't mean to say this in any negative way, it would be kind of like a blind person taking his or her fingers and gently touching your face and rubbing across your facial features and saying, oh, this is Sally. I know Sally by touching the contours of the face. That's what has to happen. B and T cells cannot identify and subsequently respond to the threat if they can't identify it. Identification means they need to touch it, okay? So here's my next question. So we're saying that identification requires that a molecule on the surface of the lymphocyte touch a molecule on the surface of a pathogen. Basically, a receptor protein on the lymphocyte has to bind to a molecule that makes up the composition of the pathogen itself. Okay. So here's my question. Okay. How does the immune system, which of course we're saying is primarily the B and T lymphocytes at this point in our conversation, how does the immune system, B and T cells, bind to a pathogen it has never seen before? Okay, so implied in my question is this other question, right? If the B and T cells have never seen a pathogen before, why should I anticipate, or is it even reasonable to anticipate, that the B and T cell has a molecule capable of binding something that's never been seen before? That seems like a very high bar, a kind of ridiculous assumption, doesn't it? So take a look at that first question. How does the immune system bind to a pathogen it's never seen before? Think about that. And if you have a response, please put it into the chat window. Okay. Okay. So thanks for the responses in the chat window. Okay. And I'm going to read my question again. How does the immune system, meaning the B and T cells, bind to a pathogen it has never seen before? And then in parentheses, doesn't it seem unreasonable to expect a B or T cell to have a molecule to something it has never seen before? Doesn't that seem unreasonable? I mean, it's kind of like this. Think about it, folks. Isn't it unreasonable for Dr. Allen to give you a study guide where he doesn't really tell you anything? Isn't that unreasonable? Well, it depends on your perspective. From yours, it's unreasonable. From mine, it's not unreasonable at all. Okay, so let me ask you a question, and this is a philosophical question. Okay, and thanks for the continued comments. I am reading them and I'm going to respond to them. Here it comes. If a teacher like myself doesn't give you a detailed study guide before an exam, your choice of responses are to give up and decide I'm not going to pass this test, or what else? What is your other option, folks? Okay, all right. Now you're seeing it, right? Your option is to figure it out yourself, to study it yourself. That's right. In other words, either give up or actually study. And that's what I'm demanding. Okay, I want you to study. But hidden in this lecture is basically the answers to everything you're going to need. I'm just not going to tell you this is the answer to the exam question. Okay? So this is the strategy of the immune system, folks. Okay, this is the strategy here of the immune system. Okay. How does the immune system bind to a pathogen it has never seen before? Doesn't it seem unreasonable to expect a B or T cell to have a molecule to something it has never seen before? Let me take a look at some of your answers. Chelsea, are there certain regions on the outside of the pathogen that they are attracted to? Well, possibly, but to be attracted to these, Chelsea, they would need to have a specific molecule. So we get back to our original question. Why should I anticipate that I have this special molecule? I've never seen the pathogen. Why should I be prepared to engage it? Okay, Yasmin, you say it has to match the receptor. Well, again, why could it match the receptor? Okay. Kit, you say the immune system maybe was exposed to something similar enough beforehand. Kit, there is some value in what you're saying. There is, because there is such a thing as immunological memory that does exist. So in some cases, what you're saying bears out to be true. And there are documented cases in medical research where getting a vaccine against one pathogen seems to offer some cross protection against a second different pathogen. There is some truth to that. Okay, so that's a nice idea. But that's not what I'm fishing for. Here's what I'm fishing for, and we're going to move forward. If your immune system has truly never seen this pathogen before or anything like it, the immune system either gives up, which means the immune system cannot identify, cannot adapt, and therefore half of your body defense is broken. And if the other half can't do its job, the pathogen will overcome you and it will kill you. That's the reality, folks. Or the immune system can be better prepared. It can study more. Like a good microbiology student, it can study more. Here's what happens. And I'm going to give it one more question in the chat window. Here it comes. Here it comes. Is it mathematically possible to ensure you win the Powerball lottery by purchasing a ticket with every possible number combination that could be drawn by the lottery? Is that possible? I mean, is there a finite number of number combinations that you can purchase a ticket with? And the answer is yes. Okay. Yeah, Kit, you would need a lot of money. So that's not a really good strategy to win the lottery, but it is possible. And this is what I'm suggesting here with your immune system. Your immune system prepares to win the lottery, not because it's ever seen the pathogen before and knows what to expect, but because it hasn't. And so before you're infected, the genes inside your immune system cells are swapping DNA segments around to build random proteins that get put on the outside surface of your B and T cells. So your body manufactures an army of slightly different B lymphocytes and T lymphocytes that have randomized proteins on their surface with random shapes, so that hopefully when your body encounters a pathogen that it has never seen before, you will have a B cell or T cell with a randomly shaped receptor capable of binding something that's never been seen before. So Kit, your question in the chat window. So it's possible our immune system builds enough random types of proteins / receptors on the surface so that one of them is the right one to react to the threat. Yes, Kit, that's exactly what your immune system does for you. Okay, it does for all of us. Okay, and so let's document the answer to this question. Your immune system, before you are ever infected, works to manufacture an army of B and T lymphocytes. Okay. Each member of the B and T lymphocyte army is a little bit different from the perspective that it's going to have some different proteins on its surface. So each member of the B and T cell, quote, army is a little different because rearrangement of gene segments in the cell's DNA ensure that different proteins are eventually made and put out on the surface. Placed outside the plasma membrane, where these different proteins have the potential to bind with foreign molecules on the surface of pathogens that have never been seen before. We'll have a molecule produced randomly capable of binding a threat, even if the threat has never been seen before. Okay, this is the crux of question 4. How does the immune system bind to a pathogen it's never seen before? Well, before that infection takes place, your immune system is building armies of B and T lymphocytes and programming different proteins on the surface of these B lymphocytes and T lymphocytes by randomly swapping segments of DNA which codes for different proteins. Okay, so the process of DNA rearrangement being referenced above is used to build a very diverse army of receptors capable of binding pathogens and also a very diverse army of antibody proteins capable of binding pathogens. Okay, so what I'm saying is this randomization of segments of DNA inside the cells is the solution to ensuring that your B and T cells can identify pathogens. And it is also the solution for allowing the B and T cells to build antibodies to things that have never been seen before. So I'm going to show you a video which talks to you a little bit about this DNA arrangement business in a whimsical way, a silly little way, which will hope get the idea across to you what cells are actually doing. Okay, so this is the second video right here, so I'm going to show it to you at this time. Let's continue, and I'll cue this up. Okay. Here we go, folks. hold on to your hats. I'm never sure of the audio, okay? So hold on to your hats a little bit. Here we go.
People are full of antibodies. A lot of antibodies. These proteins are a vital part of the immune system. But just how the body makes such a variety of them mystified scientists for decades. The human immune system has to fight off an endless variety of bacteria, viruses, and parasites. And for each invader, it needs a specific antibody. Luckily, it can generate billions of them, each slightly different, ready for any disease. But these antibodies, made from 2 light chains and 2 heavy chains, are proteins. And billions of different proteins should need billions of genes to make them. Humans have around 20,000 genes. The solution to this problem was a Nobel Prize-winning discovery. Antibodies are made by B cells. Like every other cell, B cells start off with the same 20,000 genes. But they also have an enzyme called RAG. RAG is a DNA shuffler. It targets the genes that make the antibody's binding site. There are 3 kinds of gene segments here, called V, D, and J. For the heavy chain of the antibody, humans usually have around 130 different V segments, 27 D segments, and 6 J segments. The RAG enzyme binds to one of the J segments at random, and then to one of the D segments, chopping off the spare DNA between them. Other enzymes then join the ends together. The same thing happens with a random V segment. So you end up with a gene composed of 1 V, 1 D, and 1 J joined together. This provides over 20,000 possible VDJ combinations for the heavy chain. The light chain can have another 400 possible gene combinations. Combining heavy and light chains gives approximately 8 million different possible antibodies, each with a different binding site. And that's not all. During the joining, extra DNA is added or removed at the junctions of the segments, which takes our antibody diversity from the millions into the billions. This is how our immune system can respond to diseases it's never even seen before. In fact, there's so much diversity that one of our antibodies could potentially bind to almost any target in the whole universe.
Okay, folks. So, a little bit of a silly approach to it, but hopefully it gets the idea across. As far as I know, there's no video game music playing inside the middle of your immune system. But hey, why not for the last day of class? So, okay, I don't want to belabor a dead horse on this, but let me just ask, do you have any questions about the topic we've been talking about right now? Are you okay? Anything you'd like to ask about? Okay, well, thank you for the feedback, Yasmin. So let's continue here. All right. And so what I'm going to do now, there's a lot of content in chapter 17, but you do have a week until your final exam, so it's not unreasonable to ask that you read it and study it. What I'm going to do now is take you through some of the typical steps of an immune response and highlight some various features of this process that intersect with the content of chapter 17, that is assigned reading. Okay, so I'm going to try to give you a little bit more support here to understand some of the things that you're going to encounter during your reading time. I will say I'm not going to run this advanced video for you in class today, but I really think, particularly after you've done a little bit of studying, you really might enjoy watching this advanced video on your own at home. I think some things will start to click and it'll make some sense about things. Okay, so I want to encourage you to do that when you're able to. Okay, so I want to do quite a bit of drawing, so I'm going to insert a brand new page here and just start drawing rather than typing I need to draw. So we're going to do, steps in a typical immune response here. And rather than typing the response out, I'm going to draw it out, okay? So I encourage you to draw right along with me. If you have some colored pencils at home, some colored pens, it can be helpful to do this kind of thing. Okay. So as the old expression goes, there's more than one way to skin a cat, right? That's certainly true about, well, how does the immune system get activated and riled up? There are more than one way to do it. But I'm going to show you one common way that medical science does understand. So let's start with something foreign entering the body. And just to make it easy right now, I'll talk about virus, okay? So, and you appreciate that what I'm drawing is not drawn to scale because I just don't have the ability or the technology to do that kind of thing, okay? So let's say that we have a virus lurking in our body fluids, okay? Sorry, my technology's stuttering a little bit. Doggone it. I hope this is going to work. Okay, so here we have a virus. It has entered our body, okay? And so, we mentioned earlier that B and T cells have to be able to touch this thing in order to start a response. The reality of it is that the vast majority of our B and T cells are going to be sitting in our lymph nodes. Yeah, you can have B and T cells floating through the bloodstream, but the vast majority of them are sitting in the lymph nodes. However, non-specific defense cells like macrophages are floating around the body, all kinds of body fluids, acting as police officers walking a beat. And they're checking for problems, right? So the virus enters the body, okay? And come on, technology, it'd be nice if you would work. Oh. Okay, so number one. Oh, come on. Okay, so we'll say that we have a macrophage lurking in the vicinity of this virus. Sorry, this seems very stuttery. My digital pen is not responding too good today. All right. So you have something like a virus or a bacterium, something that's in the body that doesn't belong. The macrophage job is to survey the environment and find things that don't belong, right? The macrophage cannot identify this virus. The macrophage cannot say this is a cold virus particle. It cannot say that this is a coronavirus particle, but it can tell that it's foreign. So the macrophage will touch the surface of the virus particle and kind of scan the surface looking for something. So let me ask you in the chat window, and folks, it is 1:45, which is the traditional stopping point of lecture. So before I type in my science question, let me ask you another question. Do you need a bathroom break? Do you want me to pause for 5 minutes so you can go to the bathroom? Anybody need that break? Okay, so we're having a response of a break here. Okay, gang, let's do this. 10-minute break. 10-minute break. Let's get started at 1:55, 1:56. Okay, folks, I'm going to stop sharing my screen and I'm going to pause the recording. Recording in progress. Okay, welcome back, folks. Hope we had a chance to get a little refreshment, use the bathroom. Let's continue here with our conversation. Now, I promise not to take all of lab time. I won't do that, but I need to give you a little bit of support, okay, on this topic. So, getting back to our conversation, right? We're trying to describe one of the typical ways that your immune system can get activated and what can happen along that path, okay? So the first thing there, something foreign has to enter the body. A macrophage is going to be in the vicinity because it's a police officer walking around in the neighborhood, okay? The macrophage bumps up into the virus and kind of does a pat down of the suspect. Like a police officer might pat down a suspect, the macrophage is going to pat down the suspect. Here's my question in the chat window, folks. What is the macrophage looking for when it pats down the virus? What is it looking for? If you've had, if you've had this in other biology classes, I would anticipate that you could answer, but I appreciate, you're coming from different backgrounds. So Kit, you say something to identify it. That's not incorrect. We're simply looking for what is the thing that the macrophage is looking for. Samantha, the macrophage is not looking for a bacteria, not in this case, because we're looking for the virus, right? So Elena, you say an antigen. Elena, what kind of antigen? Okay. All right. Thanks everybody for participating. I appreciate that. And again, to move the conversation forward, this is unpleasant that we have to do this so rapidly. The macrophage is looking for a type of self-identification antigen called MHC, major histocompatibility complex. Okay, so let me continue here. Okay, number 2. Macrophage looks for MHC, major histocompatibility complex, on the surface of the virus. Okay. Folks, MHC is a type of self-identification antigen, a self-identifying antigen. In other words, most of the cells of your own body are going to have MHC on the surface of their plasma membranes. And that MHC helps the body regulate its own defense system because you can distinguish friend, meaning yourself, from foe. meaning a non-friend, something foreign, a pathogen that doesn't belong in the body. So think of it this way. M8C is kind of like the United States Army giving all of its soldiers a blue armband. And when the soldiers go out on the battlefield to fight the enemy, anytime somebody in the US Army is looking out on the battlefield with binoculars and they see somebody with a blue armband, they know they're looking at a United States soldier, and therefore they should not shoot that guy because he belongs to our team, belongs to our body. Okay, that's the blue armband. M8C is like a blue armband. Okay, so question here in the chat window. Should we expect that a foreign virus has our M8C? Okay, correct. You know, that shouldn't be happening, right? A foreign virus should by and large not have our M8C. So, you know, there are a lot of complications that can go on in reality in the real world. We don't have time to talk about them all. So I'm just going to tell you the traditional route here. Please ask questions along the way if you have them. So the macrophage, which is a white blood cell of the innate immunity defenses, is patting down this virus that entered our body, looking for M8C, which identifies friend from foe. The macrophage doesn't find any M8C, so the macrophage knows the virus is foreign because it cannot identify any M8C. The macrophage knows this object, which I'm calling a virus, is foreign. Again, the macrophage doesn't know that it's a cold virus, but it knows it's something foreign because it cannot find the M8C. Okay, so no M8C found. Therefore, the object that we're patting down, in this case, the virus, is foreign. Okay, before I go any further, any questions about that? Okay, let's keep going. So this might seem like a gross anatomy, but if I stick with the police officer analogy, right? If the macrophage cannot identify this thing and knows it is foreign, the macrophage is going to eat it. That's phagocytosis. So that would be like a police officer patting down a suspect, not locating the suspect's wallet, so it cannot identify the suspect, so the suspect is foreign, and the police officer starts to cannibalize the suspect and eat him or her. Okay, so the virus is going to enter the macrophage at this point through phagocytosis. Okay, and so there's some steps in phagocytosis that for time purposes right now, I'll just kind of ignore because I just want to tell you the general story of what's going to happen here. Okay, and so the virus is internalized because the macrophage believes the virus is foreign. The macrophage cannot identify it, but it knows it is foreign. Okay, any questions? Okay, so the virus is sitting inside the cell, and what the cell will do is it will run the virus through one of the defense organelles of the cell, which you might remember is the lysosome. So here we have the lysosome. And excuse me, I want to try to use a different color here. Oh, it'd be nice if technology worked. Okay, so here we have the lysosome. The cell will run the virus through the lysosome like a piece of wood getting funneled through a wood chipper. And what comes out the other end is fragments of the virus. Okay, so at this point, we now have viral fragments. And so I'll just draw some little squigglies that are, red colored, that kind of thing. These are viral fragments. These are pieces of the virus. Viral pieces. Sorry about that, folks. My tech is really not helping today. So viral pieces, the virus gets run through the wood chipper, so to speak. While the cell is doing that, there's other machinery inside the cell that's accomplishing a different goal. So you might recall from your time learning about the anatomy of a typical eukaryotic cell, right? You learned the lysosomes and mitochondria and all that kind of good stuff. Well, there's going to be another section of the cell where you have the rough endoplasmic reticulum, okay, the rough ER, okay? And you remember the rough ER has ribosomes on it, and ribosomes make protein. So long story short, the rough ER inside the cell is going to be manufacturing MHC proteins. Isn't that curious? The rough ER is going to make MHC proteins, which I'll color code as, let's see, I'm going to color code it as a light green, okay? I'll color code it as a, well, let's see. I guess, let me do light blue here, light blue. So we're going to make MHC proteins. Okay, and for those that have had some background, the purpose of the MHC proteins being manufactured in the lysosome is so that the cell can take the viral pieces that it created by chewing the virus up in the lysosome, and it can glue it to the surface of these MHC proteins. So these viral pieces right here are going to be moved over, and they're going to be stuck on the end of the MHC protein. Okay. All right. So let me ask you a question in the chat window next. Okay. What will the macrophage do with the viral pieces fused to the MHC protein? What's the macrophage going to do, gang? Okay, I'm going to go with Yasmin's answer first, and Kit, you're absolutely right. You're going to put these fragments on the surface of the macrophage cell itself. You're going to stick these things outside the plasma membrane, which means this cargo, which I will try to highlight next. This cargo here is going to get stuck outside here. So we have these fragments getting placed outside the macrophage. Okay. So for step 3, I'll say this: the macrophage destroys the virus, puts pieces of the virus on M8C, and puts those fragments outside the cell. Okay, so that's kind of what we've encapsulated in our drawing. So from start to finish, the virus entered the body. It is foreign. The macrophage is the police officer. It pats down the virus, looking for M8C, which is a self-identifying protein. Macrophage doesn't find it. So the macrophage believes that this is something foreign and potentially a threat. So the macrophage eats the virus. That's phagocytosis. The virus enters the cell, gets run through the wood chipper, which is the lysosome. So the virus is broken down into pieces. Those viral pieces are glued to the end of M8C proteins, which are being made by ribosomes on the rough endoplasmic reticulum. Those pieces then are put outside the cell. So we've summarized this whole cartoon that's before you at this time. So here's my next question. Okay, coming in the chat window. What is the purpose of placing the viral pieces fused to M8C outside the cell? What's the purpose of doing that? Why is the macrophage doing this, gang? What's the purpose? Doesn't that seem odd? I mean, after all, I just destroyed this virus particle. Surely it's not a threat anymore. Okay, so just to keep the conversation rolling, I'm going to keep moving forward. Kit, I like what you suggested in the chat window: to alert other cells and provoke an immune response. Okay. Rachel, thank you. I appreciate that. You know, it's important that you are thinking about this process. The purpose of this behavior of the macrophage is to alert other cells in the body that there is a threat in the neighborhood. That's right. Now, we don't want to just alert any old cell. The reality of it is we want to alert B and T lymphocytes. And so alerting B and T lymphocytes can happen a number of ways. And again, for time purposes, I'm going to make some I'm going to make some compromises in what I tell you, okay? But here's the basic idea. The cell that we really want to get the attention of is a T cell, particularly a category of T cell called a helper T cell. And you might ask, well, why do I want the helper T cell's attention? Because the helper T cell is the master cell of the immune system, which has ultimate authority to turn the adaptive immune system on. So if I don't get the attention of the helper T cell, any response that I might get from subordinate cells will always be of a diminished capacity. So I want a full-blown response. I want all-out war on this virus because I want to kill it, okay? So what you're seeing right here in your drawing at step number 3, and I'm just going to try to highlight this a little bit here with, I don't know, I guess with gray. I haven't used gray yet. Let's try gray. So this stuff right here, where I'm displaying the information, when the macrophage does this activity of putting this material outside the cell, the macrophage is presenting information to the immune system. And therefore, the macrophage has become an antigen presenting cell. Okay. So, this activity, and by activity, I mean putting that material outside the cell, turns the macrophage into an antigen presenting cell. I don't mean to say that it physiologically changes the cell. I mean to say that as human beings, we're defining something important about what has just happened. I'm now going to call the macrophage an antigen presenting cell because that's what it's doing. It is presenting antigens, pieces of the virus, to the outside world within the body itself, okay? This activity, come on technology, makes the macrophage and excuse me, wrong English here, and antigen presenting cell. Okay, an APC, an antigen presenting cell. And so, as I said, the whole purpose of antigen presentation is to get the attention of other cells, but the big honcho that we need to get the attention of is a helper T cell, because it has authority to activate the full-blown response of the immune system. Okay, so we are looking, hopefully, for another cell. And as I said, most of your B and T cells are going to be in your lymph nodes. So really what's going to happen, in which the video you saw alluded to, this macrophage here, well, it's going to release some chemical alarm signals called cytokines, which will act as a chemical attractant for other macrophages to go into the same neighborhood and attack other viruses that might be there. But often the case is that the macrophages can't handle the threat by itself. So this macrophage, which is presenting antigen, is going to circulate into the lymph nodes. And once it gets into the lymph nodes, it's going to go into the anatomy of the lymph nodes where the B and T cells are held, and it's going to go around like a police officer holding up suspect drawing, and it's going to ask the cells in the lymph node, hey, have you seen this guy before? Have you seen this guy before? Have you seen this guy before? So it's kind of like the macrophage has a sketch of the enemy, and the macrophage wants to find the police officer that has seen this enemy before. Of course, what we're looking for is a T cell that has seen the enemy before. And I don't mean literally it has seen it before. What I mean is the T cell has a receptor of the correct shape that combined to one of the presented viral fragments. Okay, so what's going to happen is the following. Okay, and I promise I'm gonna wrap this up soon, but it's just a big, heavy topic here. Come on, technology, hello. Okay, so let's do this. Let's mention that in the lymph node, the APC presents the sketch of the virus to be in T cells. Okay, so in the lymph node, so the macrophage in the drawing would travel through the blood supply, leave the blood supply, get into the lymphatic fluid, go up into the lymphatic fluid of a lymphatic vesicle, okay? And start showing around its sketch, its presented viral fragments. And so what we want to find is a helper T cell that has a receptor capable of binding one of these red fragments. Now, I'm not much of an artist, but let me just draw something here. Let's see, let's do it this way. I'll do purple. Okay, so let's say that I have a big helper T cell here. And it's got molecules hanging off its surface, some of which hopefully can actually dock into, technology is so irritating. Dock into the viral fragment. So hopefully you can appreciate that. That's kind of a squarish rectangle blob that could dock into the tuning fork of the viral fragment there, okay? If that event is successful, if the helper T cell is located in one of your lymph nodes that happens to have a molecule that allows it to interact with one of the displayed viral fragments, then something magical happens. Bang, the immune system is given permission by the helper T cell to fully activate. Okay, so number 4, if the helper T cell, and that's often abbreviated T subscript H, if the helper T cell can bind to the presented fragment on the APC, the immune system turns on and begins its adaptation to fight this enemy. I mean, the immune system's always kind of on at a low level, but to get the immune system riled up to defend against one specific threat, this binding event has to happen. Okay, and okay. So, thoughts or questions right now? Oh my goodness. This technology is just infuriating. Hold up here. Jeez. Sorry, folks. That was not supposed to happen. Okay. Okay. So I think I better give up the drawing because my tech is really fighting me here. So okay, come on. All right. Okay, so let me walk us through where we're at, and then I'll type out a few words onto the screen, and I think we can wrap this up in about 10 minutes, okay? So to understand how a typical immune response happens, something foreign like a virus enters the body, a non-specific defense cell, a police officer in the name of a macrophage, encounters the foreign material and pats it down looking for MHC. If MHC is not found, the macrophage will phagocytose the foreign material. In this case, we know this is a virus. The virus is run through the lysosome and broken down into pieces. The pieces are glued to MHC proteins, which are made in the macrophage and put outside the macrophage for presentation. When the macrophage does that, it's okay for me to now call the macrophage an antigen-presenting cell. Now, as the macrophage is continuing to investigate the environment and alert other macrophage to the problem neighborhood, it will continue its journey through the blood supply, ultimately into the lymph nodes, where the macrophage wants to get the attention of the B and T cells. The most important cell that needs to have the attention received is the helper T cell, because this is capable of turning the immune system fully on. Okay, that's what we have down here. So if the antigen presenting cell is capable of finding such a helper T cell in the lymph node, then good things are about to happen, which is to say we're going to get all our defenses ramped up, and we have a good chance of defeating the enemy at that point. Okay, so I'm going to continue my conversation, I hope. Oh, technology. Okay. Oh. Okay. Okay, so what happens next? Okay. What next? Okay. So what the helper T cell will do is it will send out chemical signals which give the B lymphocytes and the T lymphocytes permission to begin to specialize. So the activated helper T cell will send out chemical signals which give permission to other B and T cells to begin to specialize. Okay. So there's some additional nuance here. For example, we really only want B cells and T cells to specialize if they have the ability to recognize the virus. That means we only want the B cells and T cells that have a receptor capable of binding the virus to amplify. That process of amplifying the cells capable of identifying the virus in this case is called clonal selection. Okay, clonal selection allows B and T cells, I'm going to put this in capitals, capable of identifying the pathogen, permission to reproduce and specialize. Okay, so clonal selection, folks, is covered in chapter 17 a little bit, and it's figure 17.6. Okay, so I'm not going to go into the molecular details of clonal selection. I don't see any purpose to that right now. I mean, we're basically at the end of our rope here. But what's going to happen is that the activated helper T cell will give other cells of the immune system that can recognize the enemy permission to grow and multiply and reproduce and make many copies of themselves. So now I have a clone army, kind of like Star Wars, a clone army of B lymphocytes and a clone army of T lymphocytes that all have receptors capable of identifying the pathogen and attacking it. Okay, so what will happen is as follows. Okay, clone armies of B and T cells capable of identifying the pathogen are manufactured. From here, the immune system is going to mount a two-pronged offensive campaign against the virus. And this two-pronged attack is talked about in your textbook at numerous places, but I'm going to draw your attention to figure 1719. Figure 1719. Okay, this is a foundation figure and therefore quite significant in its importance and what you should be thinking about. Okay, here's the basic idea. Okay, the first part of the attack is going to be handled out by the antibody-mediated defense. In figure 1719, that is the left side of the diagram called the humoral defense. Okay, and during this response, the antibody-mediated defense is carried out by activated B cells. Those B cells were activated by the master helper T cell, which originally identified the pathogen presented to it from the antigen-presenting cell in the lymph node. Okay, so this is carried out by activated B cells that grow and reproduce and specialize. Okay, the special B cells that are manufactured during this process are called plasma cells Plasma cells are your antibody factories, right? Plasma cells manufacture the antibodies. And they don't just make any random antibody. They're going to make an antibody of the correct shape to bind to molecules on the surface of the pathogen. That's why this is an adaptive response. We are making a custom weapon here. Okay. So the antibody factories do their job. And at the same time, you will generate memory B cells. Memory B cells, generally speaking, they do not kill a majority of the pathogen, but their purpose is to actually live longer than the infection itself and circulate through the body in ways that if the same pathogen enters the body a second time 2 years from now, the memory B cell might bump into it during its normal travels and recognize it and activate the immune system much more rapidly than going through the traditional macrophage route. Okay, so these are long-lived cells that can recognize the same pathogen if you are exposed to it a second time in the future. Okay. So that's the antibody-mediated portion of this foundation figure. The other side of the foundation figure is the cellular response. You know, let's be honest, when we're talking about the coronavirus pandemic, everybody's talking about, you know, what are your antibody titers? Like, you know, first, are you producing antibody against coronavirus, and how many antibodies you're producing against coronavirus? And you now know from some of the videos that even if you have antibodies, it doesn't mean you're really immune to it, right? It doesn't mean that. But nobody's talking about the cell-mediated immunity part of the defense against coronavirus. And that's because that's a lot harder to measure in the lab. Okay. So the second part of the adaptive response is the cell-mediated immunity. The cell-mediated immunity is carried out by activated T cells. And again, the activated T cells were given permission by the master T cell, the master helper T cell, to grow, reproduce, and specialize. And some of the subtypes of T cells you will make are cytotoxic T cells, okay? Let me see here. Yes, cytotoxic T cells and how they kill an infected cell of the body are highlighted in figure 1713. Okay, so I'll draw your attention to that during your private study time. The whole process of the macrophage ingesting, you know, the virus and processing it and presenting information, that is shown in figure 17.12, right? So you know, I'm trying to alert you to areas of the chapter that should resonate with you when you read, because you've been listening to the conversation here. And let's see here. Okay, so I'm going to continue and wrap this up. The cytotoxic T cells, well, they will release chemicals onto infected cells of the body and basically kill them. So you can think of cytotoxic T cells as assassins for your body that have been given permission by its government to go out and kill the enemy. Okay, so these cells attack pathogens and infected human cells that have pathogens inside. Okay, you can see from figure 1713 that they will release things called perforins, right? Figure 1713 shows you at the end of that picture, it shows you perforin and lytic enzymes. These are enzymes that poke holes in infected cells and kill them. And if you kill the infected cell, in the case of a virus, you have prevented the virus from using the cell to make more copies of itself, right? So cytotoxic T cells attack pathogens and infected human cells that have pathogens inside by releasing substances like perforin. Okay, and then we will close it up here, folks. Not only do you get cytotoxic T cells made in the cell-mediated branch of the immune system, you will also get memory T cells, okay? Memory T cells are analogous to memory B cells. It's kind of like if you're in the military, you might have the military police that are part of the Army, and then you might have different military police that are part of the Air Force. And you might have different military police that are part of the Navy, right? Each branch of the armed force kind of has its own military police. That's what I'm kind of talking about here. Memory B cells are long-lasting cells from the B cell lineage. Memory T cells are long-lasting cells from the T cell lineage. Okay, but there are some differences, but for our purposes right now, basically, we can say the same thing. These are long-lived cells that can recognize the same pathogen if you're exposed to it a second time in the future and potentially jumpstart the immune system that much more rapidly to defend yourself the second time. Okay, folks. I think I'm going to stop. Okay, there's always more to say, but I think I've covered the meat of what I wanted to say with you today, all in what, 2 hours and 15 minutes or whatever, okay? So gang, let me just stop right now and ask you, do you have any questions about this chapter, or once again, any questions about final exam content?
I kind of did want to ask in regards to the 5 different types of ways that let me get my paper, that it would help, like 5 different ways. Sorry, it's on my study guide. That antibodies protect you when they bind to antigens.